Hello friends, welcome to Dr. Ossai Physiology Academy, DOPA for short. This is the place where we make learning of physiology easy, exciting, and effective. Thank you for joining me. So today we're going to be dealing with pancreatic secretion. But if you are joining us for the first time, you are new to this channel, you're especially welcome. So if you enjoy the content we share, kindly click the like button, the subscribe button, and turn on notifications so you don't get to miss any new content we drop. All right, so we'll grow the channel together. So let's get started. So pancreatic secretion, this is where the main work is done as per secretions of the gastrointestinal tract. You see, so this is the pancreas. So it's the biggest exocrine gland. Now, the pancreas is like a two-in-one gland, okay? You know, you have the endocrine pancreas. You now have the exocrine. Okay, the exocrine is the one that secretes through duct. The endocrine part is just 10%. It's the one that secretes insulin. It's a hormone, okay? Makes up 10%. This exocrine makes up 90%. The cells that secrete the exocrine, because the pancreas is where the majority of the major enzymes that do all the major work of digestion, they come from this pancreas. So it's a very, very important gland. Okay? So, like we said, you start from the gland. Know the gland, know the cells that secrete the gland, the anatomy, and so on. So this pancreatic gland, this pancreas, now it is a lot like the salivary gland. So just check the salivary secretion, you'll get an idea of what we're trying to say. In the sense that it resembles the cells there, they resemble a grape. You have this grape-like formation. Okay? Something like this. Then like the stalk. So all here, there are cells all around here. Okay? Then there are cells here. So these cells are the acinar cells. Very similar with the cells of the salivary gland. Then these cells here are called the ductal cells. Okay? So what happens is that the same principle. The acinar cells, they are the ones that secrete the enzymatic part. Remember always that we say that all these GI secretions, what they contain, water, which contains 99.5%. Okay? Then electrolytes, then enzymes. These are the three major things. So these are acinar cells. It's the enzymes, rich in different kinds of enzymes that digest all the classes of food. Carbohydrate, lipids, proteins. Okay? So they secrete that. Then these ductal cells are the ones that secrete mainly bicarbonate plus water electrolytes. So mainly what you're concerned here is bicarbonate. A lot of it. Why? Because from the stomach, the gastric secretion is highly acidic. The pH of gastric secretion is about 1.5 to 2, all right? 1.5 to 2 to 3, around that. Initially, when it's secreted, it's 0.8, but as it gets mixed with the contents of the stomach and all of that, it dilutes it a little and it becomes around 1.5 to 3, the pH. So it needs to be neutralized. Alkaline or a base neutralizes an acid. Because for the stomach, the pepsinogen there needs acid medium to be activated. It functions in acid medium. But pancreatic enzymes, they need an alkaline medium. So these ductal cells secrete a lot of bicarbonate. And remember that when we're talking about gastric secretion in part 2, we said that one of the mechanisms of the secretion of HCl is that bicarbonate goes into the vein, a lot of it. It's like two opposing forces. A lot of acid is being secreted into the lumen, then a lot of bicarbonate is being secreted into the blood, the venous blood. So a lot of those bicarbonate that causes that alkaline tide, they now go and become secreted through the pancreas, and they come from these ductal cells. Okay? This one, the enzymatic part. So that's what you need to know. So they come and they are secreted through this duct. You see it all around. 90% is made up of exocrine. So they enter this duct to enter the duodenum. We call this part the second part. The second part of the duodenum. Duodenum is the shortest part of the small intestine, about 20 cm. Okay, first part, this is coming from the stomach. So at this point, it's enlarged to open into the duodenum. That part is called the papilla. The papilla, sometimes you can also call it ampulla of Veta. Okay? It's called the ampulla or papilla of Vata. So around that ampulla of Vata, it has a sphincter called the sphincter of Oddi. Sphincter of Oddi regulates the secretion, pancreatic secretion. So that is that. Now, we'll now go into each and every one of the enzymes. Okay? Because that will now tell us the functions. The components of that pancreatic secretion will naturally just tell you the function. So let's just go straight to the enzyme. Now, we said that the pancreatic secretion is what does the main work of digestion. It digests all the classes of food to a very large extent. So when you think about the classes of food, you think about the function, okay, and the type of enzymes. So we start with protein. So it has proteolytic enzyme. Lytic means breaks, so lyse. So it has proteolytic enzyme. So example of proteolytic enzyme, the main one there, very abundant, is called trypsin. Trypsin. We also have one called chymo. Trypsin. There are so many. Okay, you don't need to. But elastase, you have nuclease, and so on and so forth. Carboxypeptidase, and so on. So now we are done with protein. It has all this protein. You see all the enzymes just for protein that it has. Number 2, carbohydrates. So it has amylolytic enzymes. Amylolytic enzymes. Okay? So what you have there is, we call it pancreatic amylase. It's called pancreatic. No, for the salivary one, you say salivary amylase. So this one is pancreatic amylase. We break down carbohydrates. So protein, carbohydrates, next one is fats or lipids. So lipolytic enzymes. An example of a lipolytic enzyme is pancreatic lipase. Okay? Pancreatic lipase. There's another one called colipase. There's another one called phospholipase. A and B. Okay? So these are 3 major enzymes And of course, the function is very clear. Digestion of protein, digestion of carbohydrate, digestion of lipids, fats. So these are all the enzymes. Very elaborate. So many enzymes doing so many jobs. So the next thing I want to talk about, very important for you to know, is how this trypsin is activated. It has a special way it is activated. Very important for you to know. Okay? So now, because proteolytic enzymes, they can be very dangerous. So there's a protective mechanism that the body employs. You know, like I always say, the body is made up of proteins. So when they are secreted, they are always secreted in the inactive form. So this thing you are seeing here now is trypsinogen. When it's secreted, it's trypsinogen, chymotrypsinogen, and so on. They are always secreted in their inactive form. Because if they are secreted in the active form, what do you think it will do? It will digest this pancreas as it's coming out. It's digesting the pancreas itself, because pancreas is the substance of it is made up of protein, and will lead to what is called pancreatitis. Okay? So what happens is that when trypsinogen is secreted into the duodenum, in this second part of the duodenum, there's an enzyme known as enterokinase. Okay? Enterokinase. So this enterokinase, what does it do? It's secreted by the duodenal cells, mucosal cells. It activates trypsinogen. So it turns it from trypsinogen to trypsin. So the function of enterokinase is to convert trypsinogen to the active trypsin. Then active trypsin further goes and activates its brothers or sisters. Okay? So trypsin is also an activator. It activates other inactive enzymes and goes to even activate other trypsinogens. You see, so it's now like a positive feedback mechanism. Trypsin goes back to itself, trypsinogen, to activate other trypsinogen that have not been activated. But the first thing that starts it is enterokinase. Okay, it's also known as enteropeptidase too. Enterokinase, enteropeptidase, anyone. The same thing. So, another additional safety factor for this trypsin, because it can be very dangerous, they are very powerful proteolytic enzyme can destroy, is that as trypsin is being secreted, there is a trypsin inhibitor. Trypsin inhibitor that is secreted along with trypsinogen. You see? So as the acinar cells are secreting trypsinogen, there's also secreting trypsin inhibitor or trypsinogen inhibitor, so that there is no chance at all that it will be activated as it's moving along those ducts, even to destroy it. And you have a very big problem. Okay? So that is that. So next thing you now talk about is the regulation. Okay? The regulation of pancreatic secretion. So, like I said, pancreatic secretions, apart from the salivary that is only nervous, pancreas, it's stimulated, it's controlled by nervous mechanism and also hormonal mechanism. And it's the vagus nerve also. Okay? The vagus nerve controls 75%, which is the parasympathetic nerve. Sympathetic inhibits. That is very easy to understand. What are the things that can stimulate it? What are the hormones, the GI hormones that can stimulate or inhibit pancreatic secretion. So one key hormone is called cholecystokinin. It's secreted by the intestinal mucosa. Okay, intestinal mucosa cells. It's called CCK for short. There's another one, number 2, it's number 1, called secretin, also by the intestinal mucosa cells. So these two, they stimulate pancreatic secretion, but there's a difference in what they do, major difference. This cholecystokinin stimulates the enzymatic part of the secretion. So that means it stimulates the what? Acinar cells more. Stimulates the acinar cells where the enzymatic component of pancreatic secretion comes from. All those enzymes stimulate. Why this secretin stimulates the ductal cells more, which secretes that HCO3 bicarbonate and electrolytes. So the watery component is stimulated by secretin, while the enzyme-rich component is stimulated by cholecystokinin. Okay, these are 2 major hormones you need to know. Pancreatic secretion. So there are also 3 phases similar to gastric secretion, but the only difference is their percentage contribution. You have the cephalic phase of stimulation, you have the gastric phase of stimulation, and you have the intestinal phase of stimulation. So in other words, through the sight of food, the smell of food, and the taste of food in your mouth, pancreatic secretions, they start being secreted, they are stimulated. Okay? So for the cephalic phase, number 1, the cephalic phase is 20%. It's 20% in gastric, it's also 20% here. Then the gastric phase, they now exchange. So the gastric phase here becomes 10%. 10% of pancreatic secretion starts secreting as a result of food entering the stomach. So add these 2 together, you already have 30% pancreatic secretion even before the food enters the pancreas. So the intestinal phase of stimulation is what? 70. So when the food enters the intestine, that's when bulk of stimulation comes. And when the food enters the intestine, how it does is that it now activates cholecystokinin that stimulates the acinar cells and activates secretin, because they come from the intestinal mucosa cells. So the presence of food stimulates this enzyme secretion. This hormonal secretion that now stimulates the enzyme secretion. So that is how it is controlled. When food leaves, it's no longer stimulated. I know chemical stimulation, mechanical stimulation, we talked about all this. So this is very easy to understand, very straightforward. Okay, pancreatic secretion. So for further reading, I've written a book on gastrointestinal physiology. It's a gastrointestinal physiology broken down for you. So you can get the book when you check the description box, you see a link there, and you can take it to Okada Books, you can download the soft copy just for a token. And there are other books, blood physiology, about 7 of them as at the time of this record. 7 books in physiology that I've managed to write. Good, easy to read. So get them and to help you understand this more, other little details that might not be here. Okay? So you can also check out our website. The link is also there in the description box, so that you see all that things we are doing to make the learning of physiology easy, exciting, and effective. See you in the next video.